It takes future-focused vision to find ideas that become solutions to the world's most pressing concerns. Innovation and creativity are the sparks that ignite to transform lives and society, and these need space to grow.
We are home to these creative and innovative sparks. Our work goes beyond the disciplines, across our 9 faculties, 7 campuses, and being one of Africa's top business schools. As an African global university, we stand proudly among the world's top universities. We remain at the forefront of universal discoveries and provide authentically African solutions to the world's greatest challenges and opportunities. We are pioneers in our fields, from the humanities to cutting-edge technologies, continually seeking ways to improve the world around us. Our hybrid learning systems provide quality education that enables graduates to become adaptable game changers. We have molded nearly 500,000 alumni who have made their mark locally and internationally, and we continue to inspire and equip our students to uphold our firmly rooted culture of excellence. At the heart of what we do is our people. They are creative, critical thinkers who can shape society for the better, because that is how we teach, learn, innovate, impact, and live. We are the University of Pretoria.
Dear guests, good evening and welcome to this evening's expert lecture by Professor Alan Guthrie as part of UP Expert Lecture Series. I am Prof. Vinny Naidu, Dean of the Faculty of Veterinary Science, and I will be this evening's Program Director. It gives me great pleasure to introduce Alan. Alan has been a faculty member for over 30 years and has been a major leader in the equine industry, both the commercial industry and general horse health. While he has been active with various equine health issues over the years, his pioneering research in African horse sickness has been most prominent in both scientific impact, but also in its major benefit to the country, which has allowed the country to participate in international equine events and sales. Besides the positive impact his research has had on the SA economy, his horse sickness research has also enhanced the diagnosis of the disease and started the process of introducing a new vaccine against a devastating disease. For those who have been following the local press, you would know the devastating impact horse sickness is having on horses in the country, with the constraints in the local manufacturer not being able to supply the vaccines in the number needed over a number of years now. Also important to consider will be the future impact of climate change and the movement of this devastating disease to areas in which it has not occurred before as vectors move, which is why a new vaccine and new vaccine technology is actually critical to manage this disease. So I really look forward to hearing Prof. Guthrie's lecture today on his experience in using molecular technology to both enhance animal health and develop this new vaccine. With that, I would like to invite Prof. Ferris to do the welcome message.
Thank you, Vinny, and good evening to everyone. Allow me to just open my speech. It's really wonderful to welcome all of you here this evening. And of course, I want to acknowledge the presence in particular of the Faculty of Veterinary Science, Professor Vinny Naidu and his senior management team, as well as other members of the senior management team. I've noted the Deputy Dean of Health Sciences here this evening as well. We have, of course, the Director of the Department of the Department of Institutional Advancement. Thank you. But I think most importantly, it's not just the UP event this evening. We have esteemed guests here this evening from industry, from government, from non-profit organizations. And of course, we have Prof. Guthrie and his family and friends. So please feel welcome here this evening. So for UP, this expert lecture series provides a very public platform, as you can hear from the guest here this evening, to engage with a more broader audience on the kind of research that we do and the significant impact that our research is making and the contribution, I think most importantly, to this country and the borders beyond our country. And so the expert lecture presented by Professor Alan Guthrie, who's also the director of the Equine Research Center in the Faculty of Veterinary Sciences here at the university, will focus, as you can see behind me, on harnessing molecular techniques to advance horse health. Now, molecular techniques, as Professor Naidu has also indicated, is now revolutionizing horse management and horse health. And it is providing practical solutions to complex problems in the various horse disciplines, both locally and internationally. Molecular-based parentage verification systems were standardized internationally in the 1990s and are now widely applied in most horse breeds. These systems include tests for genetic disease and performance traits. More recently, molecular tests have been developed and standardized through the World Organization for Animal Health for certification of horses as free of various high-impact infectious diseases. The diagnostic accuracy and short processing time associated with these techniques has resulted in a significant reduction in quarantine periods required for the international movement of horses, thereby facilitating international trade but also competitions. So the rapid implementation of appropriate molecular diagnostic testing systems contributes to increased efficiency in diagnosing and controlling high-impact transboundary horse diseases, as was mentioned by the Dean. So this presentation provides an overview of those techniques routinely applied in horse health and horse management, focusing on very localized South African issues and concerns, and highlights the benefits to the respective horse disciplines. The presentation further highlights how modern molecular techniques can provide solutions to develop next-generation vaccines for controlling these devastating horse diseases. Professor, we are looking forward to your lecture, and please assist me in welcoming Professor Guthrie.
Professor Ferris, thank you very much for the very kind words of introduction. Honored guests from industry, government, state veterinary departments, from non-governmental organizations, and students and horse lovers. It's wonderful to have you here this evening and to be able to give you a little bit of information on some of the things that we've done over the last few years in South Africa, particularly with horses and the molecular side of things and what that meant to us. So I'm going to start off talking a little bit about horse genotyping and what got us involved with that, which started us down the road of molecular techniques in horses at the veterinary faculty. Then going to talk about an outbreak of disease we had in 2003, equine influenza, and some of the things it introduced us to and headed us into relationships with people which have helped us go down the road. And then I'm going to circle back to African horse sickness, touch on the disease itself, a little bit about the diagnostics, vaccines, where they come from, what we've got, hopefully where they're going, and then spend a little bit of time saying where I think we may be heading in the future. As far as horse genotyping is concerned, with competition horses, the value of those animals is directly proportional to their breeding. And so it means they have an inherent value at a relatively low age. And you need to know that when people put them in a sales catalogue, they're actually the horse that they claim they are. Secondly, in sports like horse racing, where you have people wagering on the events, it's probably not only horse racing that they're wagering on, but it's one of the things that survives off wagering. The integrity of that whole sport depends on the identification of the horse and making sure you've got the right horse actually participating in the race. And it's one of the reasons we got involved in this a few years ago. If we take thoroughbred horses, so that's race horses, thoroughbred horses are the only breed of animal in the world where every single horse has to be parentage verified before it can actually be entered in the stud book of any one of the international stud books around the world. So that is put in place by the International Stud Book Committee, who set the rules for what is required. And so now, obviously, they require parentage verification for registration, and then they also require ways of identifying them. And the International Stud Book Committee says that each horse has to be identified in 3 different ways, one of them being a DNA profile. So genotyping in South Africa. Horses have a lot of different blood groups. So the initial way of doing the identification of horses was on blood typing. The International Society of Animal Genetics standardized the horse blood typing test in the 1960s. They set up an international method to report these results. And they set up international comparison tests so that laboratories doing these tests all over the world could actually report results which are equivalent. And why is this important? It is not at all uncommon that a mare will be covered in one country. She will then be transported to another country. She will foal down. For her foal to be parentage verified, you're obviously going to need the blood typing certificate of the father to show that you've got the right foal on the ground. So this was all put in place so that these could be compared. Interesting enough, this was implemented in South Africa by Professor Dieter Osterhoff, who was at our faculty, more than 60 years ago. He was one of the founding members of the International Society of Animal Genetics, and he actually started the Veterinary Genetics Laboratory at the Faculty of Veterinary Science. The National Horse Racing Authority of Southern Africa, and I know Joseph's here today, it is Southern Africa, because believe it or not, there is still horse racing in Zimbabwe, and Zimbabwe racing falls under the National Horse Racing Authority of Southern Africa, and it always has done. So the National Horse Racing Authority has had a relationship with the university through its veterinary faculty for doing parentage verification of thoroughbred horses for more than 60 years. In the 1990s, the International Society started developing a DNA test for identifying horses. In 1998, at their international meeting, they announced that the next blood typing comparison test, in other words, the 2000 blood typing comparison test, would be the last one they're organizing. The International Stud Book Committee also announced that from the 1st of January of 2002, every single horse was going to have to be parentage verified. At that stage, only about 70% of the thoroughbred foals in South Africa were being parentage verified, and there were no systems in place for doing the DNA typing. The University of Pretoria and the National Horse Racing Authority sat down and said this is something that had to be addressed, and before the end of 1998, the first genetic analyzer on campus was purchased. It was a wonderful machine, ABI 310. There may be some people, I think Michael may remember the 310. Did one sample at a time, took about 45 minutes to do the sample. So a 96-well plate took nearly 2 days to run. Yes, it did require some logistics to get a few thousand samples through that machine. In 1999, the University of Pretoria and National Horse Racing Authority set up a project through the THRIP program, which is part of, administered by the Natural Research Foundation with the Department of Trade and Industry, to set up the systems to do DNA typing of thoroughbred horses. In November of 2001, the system was declared fit for purpose, and every single thoroughbred foal born in South Africa since the 1st of January of 2002 has been parentage verified using DNA typing. A few years later, and Skulk is a little bit before your time, I think. Okay. National Horse Racing Authority were having some issues where people were saying blood samples that had been submitted to the laboratory for detecting prohibited substances, it was impossible they could have come from their horse. So the National Horse Racing Authority said, guys, is it possible to get DNA profile out of horse urine? And we said, well, it's worth a try. Unfortunately, horse urine contains a lot of different salts, particularly calcium carbonate, and that doesn't work well with a lot of the different molecular techniques. So actually had a meeting with the head of the clinical pathology laboratory, Professor Fred Reyes, and said, Fred, we've got this problem, what do we do? He said, Alan, have you tried adding acid to it? I said, no. So he said, have you got some horse urine? I said, yes. I said, bring it down tomorrow, let's have a try. Added some hydrochloric acid, get this lovely bubbles, and carbon dioxide blows off the top of it, and you got rid of the inhibiting salts. And that simple intervention has now been put into place in most racing jurisdictions around the world and routinely applied by South Africa and internationally. So that was the first step into forensics. If we look where we are today, at the veterinary faculty, the veterinary genetics laboratory has genotyping systems for almost all domestic animal species, the majority of wild animal species in this country. It developed the RODIS system, which is the rhinoceros DNA index system, which is applied both locally and internationally for rhinoceros crimes, which unfortunately still continue. They've also been involved with doing forensics, stock theft, and other forensics where the animals are either the victim or the perpetrator of a crime for various authorities, including some of the welfare organizations. Unfortunately, things don't stand still. What's happening? We're going through a transition period, and there is probably going to be a next generation. Right now, there's a trend towards genotyping by sequencing. International Society of Animal Genetics has designed panels, or is designing panels for different domestic animal species. They have started comparison tests for these. They have an advantage in that they have between 100 and 1,000 times more markers than the current systems, so give you much better definition. Not only do they include identification markers, but they also include a number of trait markers. So they may be genetically determined diseases or other traits like endurance or speed, etc., which tell you something about the performance of those animals. If we look in production animals, they are used extensively, and I think one of the more interesting traits that they're now testing in cattle is to work out which cattle produce less greenhouse gases. Other species are in transition at the moment. So horses are in transition, and the horse comparison test will be implemented this year. University of Pretoria is in a position to harness this technology and will hopefully be taking it forward in the future. What about equine influenza in South Africa? 9th of December 2003, friend and colleague from the Cape went into a training yard in Milnerton and observed a whole lot of horses with temperatures coughing. The State Veterinary Department were notified because at that stage we hadn't had equine influenza in South Africa since 1987. The diagnosis was confirmed on that date using a commercially available kit that works for all influenza A's, so whether it's human, avian, or equine. And samples were sent overseas to see what we were dealing with to the World Reference Laboratory for Equine Influenza in Newmarket, the UK. They sequenced that, and we got the results back early the next year, showing that we were dealing with a different subtype of the North American strain of equine influenza. Equine influenza is a fairly unique disease. We've had 2 outbreaks in South Africa. We haven't had a single horse die of equine influenza. But it is the most spectacular destroyer of horse events. So, as far as responsible equestrian event organizers, it is a constant problem. South Africa, the outbreak in South Africa occurred and stopped racing and other equestrian sports through most of South Africa for about 6 months with obvious impacts. So to this day, the major international sporting bodies for horses have measures in place to protect the events which, end of the day, are driving the economy of those industries to a large degree. So when it happened, like most diseases, what's the first thing you do? You stop animals moving. So they started in the Western Cape. You then start asking people, have horses moved, gone anywhere else? Yes. 2 days before it was picked up, one of the major racehorse trainers had transferred a whole bunch of his horses from Cape Town to Rheinkiesfontein here in Midrand. So yes, before we'd even worked out we had the disease, it was already brewing in Cape Town and in Gauteng. How do you control equine influenza? Vaccination. With the horse population in South Africa at that stage, about 250,000 horses, and the horses in competition, we estimated that we're going to need about 100,000 doses of vaccine. At that stage, there were 3 registered vaccines in South Africa, one produced by Onderspoort Biological Products. On the morning of the outbreak, they had 600 doses in stock. One of the more businessly orientated private practitioners, as soon as he heard a whisper, he bought all 600 doses. I think there are veterinarians in the audience who will probably know who the person was and is. As far as other vaccines were concerned, there was another one available, registered in South Africa. There were 16,000 doses of it. Those were imported. Third vaccine, there were none available worldwide. So we had a total of 16,600 vaccines in South Africa at that time. So the veterinary authorities said, well, what's available internationally? And they needed to meet the World Organization for Animal Health requirements for equine influenza, which there are, and 3 were identified. So they gave emergency approval of the vaccines. 3 were authorized for importation. The only company who actually managed to deliver was Merial. Merial have since been, and I don't know what the right term to use is, Merial are now Boehringer Ingelheim. Okay. I don't know how they went from one to the other. Okay. And they supplied us with 120,000 doses of Protec flu vaccine. This is an interesting vaccine. It's a genetically modified organism where you've got a canary pox virus into which they have put the genes of equine influenza. It had just been launched in Europe, and they had a large, and licensed in Europe, and they had a large number of doses in stock and fortuitously could supply it to us. The other interesting thing about it, one huge advantage of this platform and other platforms is that you can differentiate infected from vaccinated animals. So it gives you DIVA, which helps you in your outbreak control and management. The last case occurred in March of 2004. Interesting enough, and no, we didn't pass on equine influenza to our good friends in Australia. They managed to get it themselves. And they had an outbreak in 2007. Based on our experiences in South Africa and the use of the Protec flu and the advantage it gave them, it was actually the only vaccine they considered for use in their outbreak in 2007. So here's a picture of the DIVA side of it, where you've got, when you vaccinate with a recombinant canary pox vaccine, you get a certain picture with the tests, whereas if it's naturally infected or you use other vaccines, you can't tell the difference between the two. The international requirements for equine influenza vaccines to this date include a strain representing Florida clade 2. There are 2 virus strains which recommend those or fulfill that. South Africa, number 4 of 2003, and Ohio 2003-like viruses. So those viruses, or the sequence of the South African equine influenza outbreak, now forms part of equine influenza vaccines available worldwide. Also, Richmond 2007 is included in ones that are approved internationally. All right. What about African horse sickness? First is neither horses nor donkeys are native to southern Africa. They were introduced in the 1600s. No horses nor donkeys traveled down Africa when people migrated from North Africa to southern Africa. So the first horses arrived in South Africa in 1653 from Java, with boats coming back from Java. First recorded outbreak of African horse sickness in southern Africa was in 1717. And it's when people from the Cape Colony started going inland and going on hunting trips elsewhere in South Africa. The virus is, its natural host is actually the zebra. So it had been living in Africa with zebra for many, many years. And if you think about back then, if you were a self-respecting zebra and you happen to be infected with African horse sickness virus and it slowed you down a little bit that evening, you'd probably end up as dinner. So there was lots of selection pressure on zebras to be not susceptible to disease due to African horse sickness, whereas horses, and to a lesser degree donkeys, can be infected with horse sickness. In case of horses, show severe clinical signs. In case of donkeys, show very, very mild clinical signs, or so the textbooks say. The worst outbreak of African horse sickness in South Africa in 1854-55 killed in excess of 50% of all the horses in the Cape Colony. There have been major international spreads of horse sickness. In 1960, horse sickness spread from Africa through the Middle East, went as far as Cyprus, to the west, and as far as Bombay in India, to the east. In excess of 300,000 animals died during that outbreak. And the majority of the animals involved were actually animals involved in informal sectors and animals providing transport and working animals for those people in those areas. So not only did it have an impact on the animals, it also had a major impact on the livelihoods of a number of people. Also outbreaks in Europe in 1966 and 1987. Then a more recent one was in Thailand in 2020. Just at the start of COVID. Thailand, horse sickness outbreak. Thailand had a horse population of 12,000 horses. Just over 600 of their horses were infected. So about 5% of the horse population in Thailand was infected. Then the most devastating thing about African horse sickness is of those, 564 horses died. 93% of the horses infected with horse sickness died. We've got a number of infectious diseases people here this evening. There are not many other infectious agents that give a case fatality rate of 90-odd%. So it is probably the most devastating disease known to us. Interestingly enough, and this is where I just hope there's not a third time. The European outbreak in 1987 was caused by the King of Spain liking zebras when he visited Namibia and wanting to take zebras back to Spain. That's how they introduced it. 2020 in Thailand. The reason it went to Thailand is that on the 17th of January 2020, 65 zebra, which were quarantined out near Hartbeespoort Dam, quarantined being a very loose term there, they were put on a Boeing 747 along with 74 giraffe and flown to Thailand. And they took African horse sickness with them. I hope people read the textbooks and there's not a third time, but if there is one, it's probably going to be something as silly as that. Someone wanting something to happen. The World Organization of Animal Health has played a fairly significant role. Back in the 1960s, when they had the 1960 outbreak of horse sickness, the World Organization for Animal Health said, if you are a country which is free of African horse sickness, you're entitled to refuse imports from a country that's infected. So in 1960, prior to 1960, South Africa had supplied lots of horses around the world, but suddenly in 1960 it came to an end. Since then, the World Organization of Animal Health has introduced a system whereby countries can be officially declared as free of African horse sickness. There's the map. You'll notice that there are a few countries in North Africa which are currently free, nothing in Central or Southern Africa, and then across Europe there are quite a lot of free countries. So it does still play quite an important role in the international movement of horses. What about molecular diagnostics? In the late 1990s, we looked at strategic things that we should possibly be doing in the future, and we happened to have quite a lot of material stored in freezers, and we said what we should probably be doing is developing a molecular test that can detect African horse sickness virus, group viruses. There are 9 separate viruses, 9 types within the African horse sickness virus group. So we said, okay, now if we want to detect all of them, how have we got to do it? So we went in, and Professor Peter Howell, who was part of our team, provided us with 150 viruses of different types. And then we said, okay, let's go and sequence one of the genes in there that's quite variable between the different types. And we came up with 50 of those viruses which were typed and had unique sequences for the NS3 gene. So we thought from that, if we could find conserved regions in those viruses, it could work. We then went in and sequenced 3 conserved genes in those viruses, and then identified a conserved region within those genes to aim the test at. 2 potential tests were identified, and then we started the process of validation of a diagnostic test under the guidelines of the World Organization for Animal Health. And that test was included in the manual of the World Organization of Animal Health in 2016 as one of the 2 tests which could be accepted for international trade purposes to certify animals as being free of African horse sickness, or for the diagnosis of African horse sickness if you suspect a case of African horse sickness. Obviously, we mentioned the type. The immunity of horses to African horse sickness is type specific. So you've actually, when you're developing a vaccine for African horse sickness, you're not doing one vaccine. You're actually doing 9 separate vaccines, which you're putting together to protect against all 9 types. When you have an outbreak of African horse sickness and you're wanting to possibly vaccinate, it's vitally important to know which strain you're dealing with or type you're dealing with, because they're in different bottles of the vaccines that are available. So we've also went and looked at type-specific tests using sequences that were available in the public domain, looked at the sequences of virus protein 2, which is type-specific protein, and looked at them per virus, then looked at conserved regions and characterized tests for all 9 types. Obviously, application is typing a virus during outbreak, and secondly, rational vaccination programs. Exports from South Africa. 1960, African horse sickness in the Middle East, basically, World Organization of Animal Health said, sorry, horses can't go from South Africa, or people can refuse taking horses from South Africa. In 1995, a workshop was held at the veterinary faculty, including lots of people from the state, various industry representatives, veterinarians and scientists, and with a specific aim of trying to open up a protocol for the export of horses to Europe, the EU. In December 1996, the South African delegation went to the EU and presented our case. The EU obviously thought what we had to say made some sense, and they accepted the protocol for export of horses from South Africa to the EU in January of 1997. In April of 1997, the first group of 3 horses left South Africa, heading to the EU, and for 2 of them to Hong Kong. And what's even more satisfying, one of the horses that went on to Hong Kong was a horse called London News. He went to compete at the Hong Kong International Races, and he won the 1997 QE2 Cup in Hong Kong. The other one horse that went with him was a horse called Trojan Hero, who went as his training partner. And the third horse was actually a horse called Ruby Mountain, was ridden by Kim Robson, and he was the first South African participant in the Volvo World Cup for show jumping. Now, what about African horse sickness outbreak investigations? For the exports of horses from South Africa, we have an area in the Western Cape designated for exports, and around it we have an area where we try to make sure we don't have African horse sickness. In 1999, 2 years after the protocol opened, we had clinical cases of what we thought were horse sickness. Diagnosis was done using viral isolation, which was the method of choice at that time. Took 8 days to confirm we had African horse sickness. Obviously, control measures had already been put in place. And took a further 7 days to work out that we were dealing with type 7. So 17 days until we could make the decision on 18 days, which vaccine was possibly the most appropriate to use. Unfortunately, the decision on which bottle of vaccine had been made about 2 weeks before. And it was decided to use bottle 1 because that's the viruses that were circulating that year. Bottle 7 is in bottle 2. So, in fact, time was wasted with that outbreak. In 2004, there's another outbreak in Stellenbosch. This time it took us 7 days to confirm horse sickness, another 9 days to type it, and it was type 1. In that case, we flipped the coin correctly, and bottle 1 was used and happened to be bottle 1 virus. 2011, outbreak in Mummery. By this time, real-time PCR was available for African horse sickness. We confirmed it on the day we got the samples. And then we actually went in and we did some sequencing, and we confirmed we were dealing with type 1 4 days later. So vaccination was only started after 1, confirming horse sickness, and 2, actually having got some sequence data which said we're dealing with type 1, so we could choose the right bottle. The 2 subsequent outbreaks in that area, 2014 and 2016, we now had type-specific assays, and we confirmed African horse sickness and the type on the day that we actually got the samples. So it's made a big difference to how one can actually deal with these conditions, and reduced the amount of time taken to be able to make critical decisions to manage outbreaks. The million-dollar question is, where do those outbreaks come from? 1999, it was a horse called Bless. And Bless was transported to Stellenbosch to pull a cart around Stellenbosch for tourism purposes. But they neglected to follow the protocol for movement. He come from Frankfurt in the Free State. They'd been having cases in that area. He moved down to Stellenbosch and took African horse sickness with him. So that first one in 1999 was an illegal movement of a horse. 2004, people tried everywhere. There were lots of conspiracy theories on what had happened, etc., etc. We couldn't work out where it came from. 2011, same story. Couldn't work out where it came from. 2014, initially the source wasn't determined, but looking at the clinical signs, I can remember standing on the farm with 2 people who are previous students of mine and now actively involved in the industry. And they said to me, Alan, you think this is vaccine, don't you? I said, I think vaccine's playing a role, yes. Okay. So, things had moved on. We're now at the stage where you can do whole genome sequencing of horse sickness viruses. We went and pulled, and that's one thing. I don't know how you persuade administrators at universities that you have to have deep freezers to keep stuff in the long term. Because we could go back, we could pull out samples from 2004, samples from 2011, samples from 2014, and sequence them. So that is the absolute gold mine as far as this sort of work is concerned, having the historic material to be able to go back to. We also put in the reference strains and the vaccine viruses and basically showed what had happened is that in the January of 2014, on a farm right next door to where the outbreak was, they'd vaccinated a number of young horses. They hadn't seen any problems in those horses, but about 4 weeks later, you had horses on a neighboring property infected, showing very, very mild infectious clinical signs, but from which we isolated African horse sickness. Because of the mild clinical signs, even though we knew what type it was, vaccination wasn't used as part of the control of that outbreak because we thought it was vaccine virus. The impact of outbreaks. 1999, EU automatically shuts down exports for 2 years. Exports were resumed in 2001. 2007, it took the EU slightly longer. Their bureaucratic processes had slowed down a bit, and it took till early 2007. The Mummery outbreak, we have not been able to export horses from South Africa since then until 2024. So that resulted in 13 years of suspension of direct exports from South Africa to Europe. Resumption of exports from South Africa. In 2018, the World Organization for Animal Health introduced revised conditions. Their conditions have always been, for African horse sickness, you put horses in quarantine for 40 days, and you do a test looking for antibodies, the beginning and the end, and you see whether there's been any change. 2018, they revised the OIE, the WOA code, and they actually put in that you could quarantine for 14 days and do a PCR test. If we look at May 2024, EU reinstated South Africa for export of horses. The protocol allows for a 14-day quarantine with a PCR test. So the quarantine period has been reduced by 50%. Oops. All right. And that is the aircraft taking out one of the batches of horses very recently. There was meant to be sound. I was a little scared of it because when it got to that point, it was pretty noisy. I see Candice Hobday was here a little earlier. I know she had an issue with a person being involved in a car accident. Candice was the person who actually was responsible for organizing this export of horses from South Africa. What about African horse sickness vaccine? How many people here have ever driven in a Model T Ford? Ford Anglia. Okay? Ford Anglia? That's an inside joke. All right. The first horse sickness vaccine developed in the 1930s is our Model T. Our vaccine we've got now is the Ford Anglia. So we have a 60-year update of a 90-year-old vaccine that we're using for horses, for horse sickness. However, if it wasn't for this vaccine, we wouldn't be here tonight. We wouldn't have horse industries like they are in this country if it wasn't for this vaccine. So it may have warts on it. People may say there are big problems with it. If we didn't have it, most of us in the industry wouldn't be in the industry because there wouldn't be an industry. Okay. If you look, some of you may have read the book Memories of a Game Ranger, Harry Wallater, when they opened up the Kruger Park. He was taken off the back of a horse because that's how they did all their patrols in the Sabi Game Reserve. Okay? In there, unless you understand it, he spoke about they used to have remounts supplied to them for their patrol horses every 4 weeks during winter and every 2 weeks during summer, because that's how long the horses lasted there. Okay. If you look now, it's interesting, they have horses living there permanently. They've got an equestrian facility in Skakuza. They live there quite happily. They're vaccinated. But they are the only leopard-proof stables I have ever seen, because that is their biggest threat. What are the issues? There's a very variable immune response, but you've got 9 viruses you're trying to deal with at one time. So you get not only a variation to each of the different types, but you get a variation between animals. So your immune response is not uniformly good. So generally people say if you've vaccinated a horse at least 3 times, you're going to have fairly good protection. But don't forget, there's no vaccine that's ever been developed that's 100% effective, 100% vaccinates. The other issue, these are modified live virus vaccines, and you can get reversion to virulence. So what that is that the vaccine virus, which has been cooled down, for whatever reason, hots up again. It's a well-known phenomenon. It's occurred with foot and mouth disease vaccines, modified live vaccines, and also with polio vaccines in human beings. So it's not unknown and obviously something to be very careful of. Then you can get reassortment. If you've got more than one virus replicating in a cell, you can get the viruses coming together. In these viruses, they've got segments of their genome and they just mix together. So if you put 2 viruses together with 10 segments, you can theoretically get 100 different variants coming out the other side. So you can get reassortment not only between vaccine strains, which has happened in the outbreaks in the Cape, but also between vaccine and field viruses. So there are regulatory authorities that are dead scared to look at using these in their countries. We also cannot vaccinate young foals. And with our foaling season, starting in August, September, maybe through to January, by the time our horse sickness season starts in February, March, and April, you've got a whole lot of foals which are between 4 and 6 months of age. They've lost the antibodies their mothers gave them. They're totally susceptible when horse sickness is at worst. So every year we get a reasonable number of, the majority of cases are young horses not vaccinated previously. And again, you can't vaccinate, you can't differentiate vaccinated from infected horses. Other vaccines, people have looked at inactivated vaccines. In Spain, outbreak in 1987, the type 4 vaccine strain from Onostrobot Biological Products was licensed. Merial inactivated that and produced a vaccine which was distributed and used during the 1987 outbreak. It has since been discontinued. Obviously, looking at the polyvalent, the multiple strain cover that's required for horse sickness, there is work ongoing at the moment, both in the United Arab Emirates and in South Africa, looking at this technology to take it forward. If we look at other side of things, we had our equine influenza outbreak in 2003. We had the vaccine we used, Protect Flu, for equine influenza, used the LVAC backbone and they invited people to come together to discuss our outbreak in South Africa at Mariel in Lyon in 2004, and the head of the R&D section asked me if I would attend. I said, yes, Jules, I'll attend on one condition. He said, what's that? I said, we can talk about African horse sickness. He said, no, perfect, yeah, that's fine. After the fact, he came back to me and said, yeah, Alan, when you said we can talk about African horse sickness, I didn't know what you were talking about. I went and pulled my virology textbook off my bookshelf, and I opened it to African horse sickness. I said, maybe this guy's got something that's worth talking about. So we had discussions and went into a partnership to develop a type 4 vaccine for African horse sickness. We showed a good immune response in horses, and in 2009 we did a study where we actually showed that we got good protective immunity, both from antibodies and from cell-mediated responses to this vaccine. We've since done a bit more work with Mariel, who's now become Boehringer Ingelheim. We've done 3 additional types. We've got similar responses. So we're, maybe 4/9 of the way down the road of proof of concept, but we've still got a long way to go. Okay. But there's a lot more support internationally for looking at these things now. There are other approaches, both locally and internationally. Reverse genetic systems have been looked at locally as well as in Europe. There have also been development of virus-like particles where they make these in different either viruses or in plants. The plant side of it has been particularly active in South Africa. And then looking also at nanoparticles, which are subunit vaccines. They're also being explored at the moment. None of these approaches have yet got to showing protective immunity in horses. They've done some work in mice. They've done some antibody responses in horses. So yes, there are a number of other people and a number of other approaches. But one of the major drawbacks on all of these, or there are a few major drawbacks, one of them is we still don't have a standardized challenge model for African horse sickness virus, so we can't standardize these approaches across the world to be able to say we're comparing apples with apples. And then a more interesting one is under the Convention for Biodiversity, there is the Nagoya Protocol, which basically says that either indigenous species or derivatives of those species can only be transported, exported, and used for commercial purposes if they fulfill the requirements of the Nagoya Protocol. This came in about 2013, 14 different countries. It is in place under our Biodiversity Act in South Africa. It gives, I think, gives a lot of scientists a very good excuse to keep things to themselves. But it's also made it very difficult for the major multinationals, because unlike human viruses and human pathogenic agents, which are exempt from Nagoya, animal viruses and animal pathological agents are not. So not only do you have to go through all the biosecurity side of things, you've got to also cover Nagoya. Not only does it include the viruses and their genes, it also includes their digital sequence information. So if you've got a sequence on GenBank and you say as a manufacturer, I want to use it, aha, when you come to commercialize it, they say, all right, fine, where did that come from? When was it isolated? Where is your material transfer agreement from the person who supplied you with the sample? And where's your benefit sharing agreement with those people who should share benefits? So it's a bit of a problem on the veterinary side. I do think it's something that from the University of Pretoria, we are in an interesting situation. It does pose a bit of a barrier to some of us, but I think if we get our act together, it could actually be a big advantage going forward to be able to get some of these things taken forward and supported by multinational companies. It was touched on vaccine availability. In 2008, a paper published by Honest Report Biological Products, scientists from Biological OBP, stated at that time we had a horse population of 270,000 and that they were distributing about 150,000 doses of vaccine a year. So we had about a 56% coverage. The first document I've got with issues of vaccine supply from OBP, or African horse sickness virus vaccine, dates back to 2014. It has continued to be a problem with supply since then. It's become more acute in the last few years. And my estimation, 2004 and the last 2 or 3 years before that, with our horse population having grown and what information we've got coming out on supply, I estimate that we're only covering between 25 and 30%. So OBP in 2008 said that vaccine coverage was insufficient. I don't think it would be inaccurate to say it is now grossly insufficient. And it is a tragedy because it has welfare issues, the horses themselves. The other thing a lot of people don't realize, horses in the rural areas play a very important role with animals on farms, and people use horses to bring animals in and kraal them at night. The years we have bad horse sickness outbreaks, the rate of stock theft increases drastically. So it plays a role in those communities as well. So it's something that needs to be looked at. I do understand that there are serious discussions between OBP and the Minister of Agriculture. It would be great to see something happen and this be reversed. I see no reason why there's a product which works, why they don't produce more. It's got a long shelf life. They can keep it for 2 or 3 years. They've just got to make people sell it if they've got it on their shelves. You ain't selling nothing if it's not on your shelf. New vaccine, there are a number of candidates under investigation. The issue with Nagoya does play a role with commercialization, and international companies are looking at it very carefully. From the date a company decides they're going to commercialize a product, you've then got to go through all the steps to license that product. It's going to take another 3 to 5 years to license that product from when the decision is made to commercialize. So it's not something that's going to happen overnight. However, if it's something like what happened with COVID, where they threw billions of dollars at it, it may accelerate the process. Diagnostics. Diagnostics works well, it works quickly, but it's important, just like with other viruses, to make sure that you're covering any variants that you didn't know existed. So you need to have ongoing evaluation of the tests that you're using so that you can be sure that they remain current. As far as the whole genome sequences of African horse sickness virus are concerned, at the moment, close to 100% of those sequences are from Southern Africa, very little from North Africa, and if there's spread of African horse sickness from North Africa into Europe, it's going to be those viruses, not our viruses, unless someone takes a zebra. Other side of it is the omics. So we've got the transcriptomics, metabolomics, proteomics, which are coming onto the scene now as a next generation in our spaces. I believe they could play an important role in understanding the disease mechanisms. If we compare why does a horse get sick, why does a donkey not get sick, one could actually start answering some of those questions. So I think it'd be some quite exciting avenues to go down. Also, if you understand the mechanism of disease, you may well be able to come back and look at the therapeutics. I know I'm asked questions by people on various treatments for African horse sickness, and they say, well, we're doing this, that, the next thing. My comment to them always is, does it make you feel good to do that? They say yes. I say, well, that's fine with me, because the only treatment for African horse sickness that I know that has any scientific support is prayer.
Alan, thank you for that lovely presentation, for taking us through an immense career of achievement and looking at how molecular diagnostics can be used to improve not just diagnostics, but clinical management of animals. What I found most interesting was how you've taken a challenge and found solutions for it, which I think is extremely impressive. And for that, I think you deserve another round of applause. Of course, you've given us a lot of food for thought, and I don't just mean zebra. So I'm sure the audience has a lot of questions. So we're going to open up for a few questions. I've just asked the audience not to use the mics in front of you. We have roving mics, and you can ask a question. And I'm going to kick off with the first question. You mentioned something very interesting, that this virus kills 90% of horses. Why have horses never developed immunity while zebra have?
If you look at the evolution of horses, they evolved basically the steppes of Russia and then went out from there. If you look at the where the different species separated, you had the branch which has zebra and donkeys on it, separated out, I can't remember how many millions of years ago, and obviously zebras and donkeys ended up mainly in Africa, okay? And then horses separated out and were evolved in the steppes of Russia. So I think it is just a case, you didn't have the virus there, so they were never exposed to the virus, and so it didn't happen. And so there's and I think it's going to be a fairly simple difference between the horse and the susceptible and a non-susceptible species. I think down the road we could be quite surprised what it is. It may be a single gene, maybe.
So genetically modified horses are the next step.
Yes, Vinny, they could be the next step. Not allowed according to the rules of the National Horse Racing Authority. But secondly, I'm damn certain that the gene, if it is a single gene responsible for susceptibility to horse sickness virus, will be right next to the go slower gene or the jump lower gene.
But let's open it up for questions for the audience.
There's one.
Thanks, Alan. So what if it's a mule?
Yeah, mules are interesting characters, and they fall in susceptibility. Well, it depends how you bred your mule. Okay? If it is a mule between a horse and an African donkey, its susceptibility is that between a horse and an African donkey. If it is between a horse and a European donkey, so the big Spanish donkeys, they are just as susceptible as horses. Okay, so you'd have them like that. Okay, so it depends on the actual hybrid and what the father was.
Alan, thanks very much for the incredible information that you've provided. I'm just wondering, in terms of the lack of vaccines that we have, the huge lack of vaccines, and the fatality rate, how is it possible that we are actually still sustaining a horse population in South Africa? How have we managed that? Thank you.
I'd hazard a guess to say the amount of money that people are spending on alternatives to vaccination are huge, even the people who are vaccinating. The problem with African horse sickness is when we have major outbreaks, and I compare it to burning fire breaks. If you live on a plot and you don't burn fire breaks or have fire breaks around your property, you're looking for problems. If you have a horse sickness population and you don't vaccinate those horses, you're looking for problems. But if you have a massive bushfire come through, your fire breaks aren't going to stop it. Okay. With African horse sickness, if we go back, the last major outbreak we had was 2007. Okay? Almost 20 years ago. And if you read the textbooks, they say we have major outbreaks about every 20 years. So next one's probably in the pipeline. The problem there is that your population around your vaccination population is getting a lot less vaccinated, and you end up with a bushfire, and it jumps into your horses. And I think it's a factor that the challenge of those horses is increased, and you get horses that get exposed to more virus. The other interesting thing is, and I know there's a person in the audience who will identify with what I'm going to say, it appears that certain horses actually attract more culicoides, midges. So if, like human beings, there's some people that the mosquitoes like to bite. It would appear with midges, there's some horses which are more attractive to culicoides. So you can get in a stable a single horse being infected and dying, whereas others aren't. So there are a whole lot of things we have no idea about. The African horse sickness, we're not only dealing with the equine hosts and the virus, but we've also got the midges that transmit it. So it's a much more complex issue. I think we've addressed quite a lot of the issues about the virus. We may be starting to address some of the issues which make it different in different horses, but we haven't really even started going down the path of what the midges play. And I think we could understand more. So yes, I think if we have a major outbreak again, it's going to have a much more, a much larger impact, even on well-vaccinated horses. So it's something that is of real concern.
So, Alan, just to further on that, so what is the impact? Are you expecting another 55% mortality?
No, I don't think so. Look, people have got a lot smarter, and a lot of people, as soon as they start hearing about cases, they start putting vector control around their horses, applying insecticides, applying insect repellents, putting other species around their stable. So midges, because unfortunately you take the midges that go and bite the horses, it's the adult female culicoides midges that need a blood meal for their eggs to ripen and for them to go and lay eggs. So they're just out to find a blood meal so they can go and lay eggs. They don't care what they feed on, and if you happen to have donkeys or a cattle or something like that, they will take the blood meal off your other species. So putting decoys around the stables could also help. Stabling does help, and if we go around this country, how many paardebergs are there? How many paardekops are there? If we go back before we had vaccine, what they used to do during times of bad horse sickness, they used to chase the horses up on the top of those hills because two things, the wind was a lot more up there, so any midges that took off would be blown away and wouldn't be able to do anything up there. The breeding grounds for midges were not very good on that sparse soil on the top of the hills. So our forefathers actually taught us all the lessons we need to know about how to deal with this, because they dealt with it for many years before we had a vaccine.
So we have time for one more question. See, Michael has
Michael.
Alan, forgive me if I haven't understood correctly, but my understanding is that the virus that is produced locally is live attenuated. Is that correct?
That's correct. Live attenuated vaccine.
So if we look at what's happened on this continent for a disease that was once eradicated but is now coming back, I'm talking about polio, the cases of polio that are beginning to re-emerge are those that have been caused by live attenuated virus which has activated itself and has now become pathogenic again. And that's really devastating, you know, when you think about the place that we were previously. And I'm just wondering about the wisdom of continuing to use a live attenuated virus when we have such incredibly powerful alternatives like mRNA vaccines, without which we wouldn't be sitting here. If you think about the great successes we had in COVID, and which require a manufacturing process which is completely different, and which can be adapted to many diseases. So is this not something we should be looking at?
Michael, it is, and people have tried to engage the people currently involved with the mRNA processes. There is a fairly large European Union project at the moment where it was hoped to include an mRNA candidate in that, but we could not gain access to the systems at that stage. So yes, it's on the radar. I think they're dealing with things in human medicine first, and the technology is probably going to come available down the road. Interesting, if you look at the other side of it, where you look at LVAC construct, for example. LVAC construct, there have been the Protec flu on the market since the early 2000s. They also licensed a West Nile virus vaccine using LVAC. They've licensed a number of vaccines for other small animals. Advantage is that is a biosafety level 1 agent, can be produced in any vaccine manufacturing plant, whereas as soon as you're working with African horse sickness, either live attenuated or inactivated, you're producing it in a BSL-3 facility. And there are not many in the world. So you're dead right. Technologies are there. I think we're going to see it. We're a little bit behind, and obviously those companies at this stage, I think the people who've got funds to fund things in human medicine are a lot more than they are in the veterinary medicine. Interesting, LVAC has gone on. There have been some candidate vaccines produced for humans. There was an HIV vaccine produced, some of the trials done here in South Africa. But it's a GMO, and there are lots of people, you mentioned GMO, they don't want to have anything to do with it. So I think what's happening there is from the veterinary side, some of those platforms are being developed and shown to be safe, mainly in companion animals, not in food animals, okay, because of connotations with the GMO side of it. So it's going to happen, and the modified live virus vaccines, well, all I can say, as a veterinarian who's had quite a lot to do with African horse sickness outbreaks, I would not like to deal with any outbreak without knowing I had a vaccine I could use, okay? Even one with warts on it, okay, in the face of an outbreak. And I think that's also one of the things that's made it a little bit more difficult for South Africa to get its protocol opened again for exports. The international community has relied on South Africa to provide vaccine when they've needed it. We can't even provide for ourselves now. And I think they look at us and they say, hell, if you can't look after yourselves, how are you ever going to help us?
Alan, thank you very much for that. We are running out of time. I'm sure there are a lot more questions, but please feel free to approach Alan during the light refreshments, and I'm sure he'll answer all the questions. Alan loves talking about horse sickness. That's one thing I do know, so he will be able to answer all your questions. But Alan, once again, thank you very much for the presentation. And I would like to invite Prof. Ferris to do the closing.
Well, all I think that is left for me to do is to, first of all, thank you, Alan. I think that was really an amazing journey. And even for those of us that do not come from the veterinary field, I do not have even a science background like myself. It was still very, very interesting. And then, of course, you talked law, and so then you drew me in. So, so I think we could probably all were able to find some reference point that made it interesting. But I think your ability to also just clarify what seems like a very complex problem to all of us has certainly contributed to making this talk very fascinating talk. And for me, it also underscored the importance of the work that we do at the faculty, and really the fact that we are providing solutions for challenges, complex challenges that stems from South Africa, that stems from the continent, and that we play a very important role in that regard. So once again, thank you. Also to the faculty, Prof. Vinnie, for really hosting us and creating the opportunity for sharing the knowledge that comes from the faculty with a wider audience. Thank you very much to the audience. Thank you for taking time out to join us here this evening, both some of our staff, students, as well as our guests from outside of UP. And then the Department of Institutional Advancement, who's organized this event this evening. In particular, Lynette, thank you for organizing this 34th lecture. And then also our service providers, sound and film and photography, and then outside refreshments. Thank you to you as well. So please join us for some light refreshments outside. Thank you and good evening.